Yo, yo, yo. Welcome to Biotech. So, genetic engineering techniques can be used to analyze and manipulate DNA and RNA. So, these are the ones we're going to talk about, including the nucleotide sequencing called Sanger sequencing. PCR, Polymerase Chain Reaction. It is widely used to rapidly make millions to billions of copies of a specific DNA sample in a test tube. So this allows a very small sample of DNA that can be collected to be amplified to large enough amounts to study in detail. PCR requires the following components: the DNA sample to be amplified, DNA primers, DNA nucleotides, and Taq polymerase. So what does this sound like so far to you? Well, to me, it sounds like the process of DNA replication. We have the primers being attached to the template, and then the polymerase, and in this case, the DNA polymerase, right, for DNA replication, going in and attaching the primers and starting the addition of nucleotides to replicate it. But for PCR, we have a Taq polymerase. So Taq is just simply a heat-tolerant bacteria, and so it's a polymerase from this type of bacteria. It functions without denaturing at very high temperatures, and we're going to see why that's important on the next slide. PCR involves repeated cycles of heating and cooling that allow the DNA to be created. So these are the basic steps of PCR, y'all. Things that you just need to know in general. So you don't need to know the names, just that it is the similar process of DNA replication. First one, at very high temperatures, denaturing the DNA. It separates the DNA strands. So what was holding them before was the, you guessed it, hydrogen bonds. So those hydrogen bonds are broken. Then during the second step, when it cools down, it's called the annealing step. It allows the primers to bind to the complementary sequences on the single-stranded template DNA. And finally, we have the extension portion where we just heat it up a little more. It again allows the Taq polymerase to extend the primers, thus creating the targeted new DNA strands. And then it repeats until you know a lot of DNA is created. So here's just a visual of what is being created. So we have our DNA sample. After one cycle, y'all can see, two cycles, y'all can see, three cycles, again, y'all can see. So the primers are still there. Gel electrophoresis. So after the DNA has been replicated to billions of copies, we can take that sample and run it through a gel electrophoresis. So gel electrophoresis separates molecules by their size and charge. So the type of molecules that can be used in electrophoresis would be the DNA, RNA, and proteins. Molecules are loaded into the wells, and you guys can see the wells right here. They're just openings in the gel. And then an electric current pulls the molecules through the gel, and you guys can tell it runs from negative to positive. The DNA will move down the gel as so because DNA, as you guys can remember, is negatively charged. So like repels like, and then this positive is attracting it. Molecules would travel in the gel at different speeds, thus taking them different distances, because it's based on the size as well as charge. With the size, smaller molecules travel faster than larger ones, and then the charge, stronger charges travel faster than weaker charged molecules. So in DNA gel electrophoresis, DNA samples are cut using restriction enzymes before being ran through a gel, producing DNA fragments of different sizes. So let's go over this. We have talked about restriction enzymes before when we were doing the bacterial transformation lab. We know that they can be cut open and the DNA can be inserted. So the restriction enzymes are these proteins that cut DNA at specific sequences. So DNA is then pulled towards the positive cathode because DNA is negatively charged. Smaller DNA fragments travel farther than the larger fragments. One application of DNA gel electrophoresis is to determine what alleles are carried by an individual, aka their genotype, or their genes. And what represents the genes, y'all? The number of base pairs. Different alleles for a gene can have different restriction sites that produce different sets of fragments, as we can see in all of our examples here. The normal hemoglobin allele produces 3 fragments when cut by the specific restriction enzyme, while the sickle cell hemoglobin allele produces only 2 fragments. So we can differentiate based on that as well. Another application of DNA gel electrophoresis is to determine a child's mother or father. A child has DNA from both parents. When restriction enzymes are used to cut that DNA up, the child's DNA will be a combination of fragments found in both the mother and the father. So these 3 individuals are being tested to see who's the father of the child. So for the mom, we see that it matches this here. This one doesn't match a mom, so it must be the DNA of a dad. So obviously not 1, but maybe 2, 3. Moving on, analyzing this. This comes from the mom, checks out. This comes from a dad, so it could either be dad 1 or 3. Then again, this fragment here for the child is a mom, so it checks out. Now this one doesn't match a mom, and we're looking for the ones that matches the most, which is dad 3. Dad 3 is the father. So let's go over bacterial transformation. It's simply introducing DNA into bacterial cells. And this ties back to the concept of recombinant DNA plasmids, and that can be, made to introduce new genes into a bacteria. So here are the steps of bacterial transformation. The first part, we have human insulin producing genes taken out, and then the plasmid DNA taken out from the bacteria. And with that plasmid, we use restriction enzymes to cut it. And then the DNA of human insulin producing genes are combined with the DNA of the plasmid. And then that plasmid with the human DNA for insulin is now inserted into the bacteria. And then the engineered bacteria multiply producing insulin. The insulin is separated and then purified to produce the human insulin that we know, and it can be used to treat patients. We know that the GFP, or the green fluorescent protein gene, was extracted from the jellyfish cells, and then it was manipulated to be put into pea glow, right? So to create that recombinant plasmid, um, again, we had to cut 2 pieces of the DNA, the A and the B, with the same restriction enzyme. And then when those DNAs are able to stick to each other, right? These are the sticky ends here that we have. They must be complementary to the ones up here. And they bind or join together with the help of DNA ligase. And this is how we create that recombinant plasmid.